Most people believe that cancer risk is written in their DNA and cannot be changed. I spent the first decade of my career believing the same thing. Then epigenetics changed everything. We now know that genes are not switches permanently locked in one position. They are regulated. Turned up, turned down, silenced, reactivated by the compounds your cells encounter every single day, including the compounds in your food. 5 of those compounds are backed by enough evidence that I discuss them with every patient over 60. Let me walk you through each one. Let me explain what epigenetics actually means, without the textbook language. Your DNA contains around 20,000 genes. At any given moment, only a fraction of those genes are active. The rest are switched off, silenced by chemical tags that sit on top of the genetic code. This regulatory layer is called the epigenome. Unlike your DNA sequence, fixed from birth, the epigenome changes constantly. It responds to your environment, your stress levels, your sleep, and yes, directly to what you eat. You may carry genes that suppress tumor growth, genes that trigger cancer cell self-destruction, genes that regulate inflammation at the cellular level. But if those genes are silenced, chemically switched off by epigenetic tags, they cannot do their job. The machinery exists, the instruction manual is there, but the power has been cut. Specific plant compounds can restore it, interacting directly with the epigenetic machinery. This is a documented molecular mechanism. Why does this become critical specifically after 60? 2 reasons. Both are relevant to everyone watching this. First, mitochondria, the energy-producing structures inside every cell. Most people don't know, mitochondria play a direct role in triggering cancer cell self-destruction. As we age, mitochondrial function declines. The signal that tells a damaged cell to destroy itself becomes weaker. Cancer cells exploit this decline. Second, cancer stem cells, a small subpopulation within a tumor that behaves completely differently from the bulk of cancer cells. They divide slowly, resist conventional treatment, and are capable of regenerating an entire tumor from a handful of surviving cells. Standard oncology has made extraordinary progress against bulk tumor populations. Cancer stem cells remain one of the most significant unsolved challenges in the field. The 5 compounds I will walk you through today address both of these mechanisms: epigenetic, mitochondrial, and cancer stem cell pathways simultaneously. This is not a list of healthy foods. This is a targeted molecular protocol built on 2 decades of oncogenetic research. Compound 1, pomegranate, and the mitochondrial signal. I'm starting here deliberately. Most lists begin with green tea or broccoli. I'm starting with pomegranate because its mechanism is most directly relevant to mitochondrial function. After 60, that mechanism comes first. Pomegranate contains a family of polyphenols called L-gitannins. When consumed, your gut microbiome converts them into molecules called urolithins. Urolithins have demonstrated the ability to activate mitophagy, the selective removal of damaged mitochondria from cells. This is the first mechanism. A cell that cannot clear its damaged mitochondria loses its ability to trigger apoptosis properly. It becomes harder to eliminate. Urolithins help restore that elimination pathway. But the mechanism doesn't stop there. The real story runs through your gut microbiome. There is a specific bacterium, Akkermansia muciniphila, that L-gitannins feed and multiply with exceptional efficiency. Akkermansia has emerged as one of the most significant bacterial strains for immune competence. Higher Akkermansia levels mean stronger responses to cancer immunotherapy. Their natural killer cell populations are more active, their systemic inflammation measurably lower. Now, the clinical number I find most compelling. A peer-reviewed study tracked men with rising PSA levels, the blood marker used to monitor prostate cancer progression. Standard PSA doubling time in this population, approximately 15 months. That is the baseline without dietary intervention. In men who consumed 8 ounces of pure pomegranate juice daily, that doubling time extended to an average of 54 months. The tumor biology had not been eliminated, but its trajectory had been fundamentally altered through diet alone. No changes to medical treatment. Practical application: 8 ounces of pure pomegranate juice in the evening. Dilute 50-50 with water if blood sugar management is a concern. Avoid commercial blends. Compound 2, purple potato. Targeting what most cancer treatments characteristically fail to reach, the cancer stem cell survival mechanism. Cancer stem cells divide slowly, They express drug efflux pumps that push chemotherapy compounds back out of the cell before they can act. They have enhanced DNA repair mechanisms. They are, by design, resistant to the tools we most commonly deploy against cancer. These cells are responsible for tumor recurrence. A patient achieves remission, then watches a tumor return, rebuilt from surviving cancer stem cells. This remains one of the central unsolved problems in modern oncology, disproportionately relevant in patients over 60. Here is where purple potatoes enter the picture. The deep purple pigment comes from anthocyanins, concentrated far above levels found in blueberries or blackberries. Research in the Journal of Nutritional Biochemistry. Anthocyanins from purple potato extract selectively induced apoptosis in cancer stem cells, in colorectal cancer models, while leaving healthy cells intact. The mechanism: downregulating survival signaling pathways that cancer stem cells depend on for persistence. This is not general antioxidant activity. This is targeted molecular interference with a specific cancer stem cell survival pathway. When cooked purple potato is cooled overnight, the starch transforms into resistant starch, passing intact into the colon to feed beneficial bacterial fermentation. Resistant starch directly supports the microbiome populations most associated with immune competence and reduced inflammation, simultaneously with the anthocyanin mechanism. Cook the potatoes the evening before. Serve them cold. Anthocyanin content: preserved. Resistant starch benefit maximized. Both mechanisms from a single preparation choice. Compound 3. Broccoli sprouts. The genetic master switch. If I were asked for the single most documented dietary compound for epigenetic cancer defense, the answer would not be controversial among my colleagues. It is sulforaphane. And its primary dietary source is not mature broccoli. It is broccoli sprouts. Sulforaphane activates a transcription factor called NRF2, a molecular key that unlocks a specific set of genes simultaneously, including tumor suppressor activity. When NRF2 is activated, it opens a coordinated response involving over 200 downstream genes. These genes already exist in your body. In many older individuals, these genes have been partially silenced through accumulated epigenetic changes over decades. Sulforaphane acts as a reactivation signal, restoring their expression. This is documented molecular mechanism, not metaphor. Sulforaphane's interaction with NRF2 is one of the most precisely characterized examples of dietary epigenetic modulation in scientific literature. Research at Johns Hopkins confirmed that 3-day-old broccoli sprouts contain up to 100 times more sulforaphane precursor than mature broccoli heads. 100 times. Not a marginal difference. A tablespoon of sprouts delivers a sulforaphane dose that a very substantial serving of mature broccoli cannot approximate. One additional detail: the stem of a mature broccoli head contains approximately twice the glucoraphanin concentration of the florets. Most people discard it entirely. If you are eating mature broccoli and discarding the stem, you are leaving the majority of the compound behind. Keep the stem. Always. Preparation matters. Sulforaphane formation is enzymatic. It requires myrosinase activity. High heat from boiling destroys this enzyme before conversion can occur. Light steaming, 3 to 4 minutes maximum, or consuming raw, preserves enzymatic activity and maximizes sulforaphane yield. Do not boil. The practical protocol: broccoli sprouts daily where accessible. Mature broccoli, stem included, lightly steamed, as a consistent secondary source. Compound 4. Papaya. Repairing the oxidative damage blueprint accumulated across decades, particularly in lung tissue. Every breath over 60, 70, 80 years carries oxidative burden, from pollution, metabolic byproducts, and for many over 60, from years of tobacco use. This oxidative damage does not disappear when a person quits smoking. It leaves a molecular signature that elevates cancer risk for years, sometimes decades, afterward. Papaya contains beta-cryptoxanthin, a carotenoid with specific affinity for pulmonary tissue. It accumulates preferentially in lung cells, functioning as antioxidant and cancer signaling modulator. It neutralizes reactive oxygen species before DNA damage can occur, and directly modulates the cancer cell signaling pathways that lung tissue cancer exploits for survival. Individuals in the highest quartile of beta-cryptoxanthin consumption showed a 31% reduction in lung cancer risk, documented even in former smokers. 31% is a substantial effect size for a dietary variable. The compound appears capable of modifying risk, even in tissue that has already sustained significant oxidative damage. Ripeness is not aesthetic, it is biochemical. Beta-cryptoxanthin concentration increases significantly as the fruit ripens. Underripe papaya contains a fraction of the compound. The mechanism involves inhibiting specific kinase signaling pathways that cancer cells in lung tissue exploit for survival and proliferation, while supporting DNA repair in healthy cells. 1 cup of ripe papaya daily, fruit with predominantly yellow to orange skin and slight pressure give, is the intake level consistent with meaningful research benefit. The seeds of papaya are edible, flavor profile resembling black pepper. Emerging research interest in cancer biology. I see no scientific reason to discard them. 2 compounds addressing the epigenetic layer. 2 addressing specific tissue vulnerabilities. The 5th addresses the infrastructure that tumors must build to survive. Compound 5, green tea. The EGCG compound that disrupts the vascular construction project that every growing tumor depends on. A tumor is not simply a mass of abnormal cells. It is, functionally, a construction project. Without new blood vessel formation, it cannot grow. To grow beyond approximately 2 millimeters, a tumor must recruit the body's own vascular system to build a dedicated blood supply. This process is called angiogenesis. The primary bioactive compound in green tea, EGCG, is one of the most extensively studied natural inhibitors of tumor angiogenesis in scientific literature. EGCG interferes with vascular endothelial growth factor signaling, the molecular pathway cancer cells use to recruit blood vessel formation. This disrupts the construction project. It simultaneously reduces nuclear factor kappa B, a master inflammatory gene regulator that cancer cells exploit to create a pro-survival environment inside the tumor. And it has demonstrated direct stimulatory effects on natural killer cell populations, a 47% increase in NK cell activity in one well-controlled study. 3 distinct mechanisms, 1 compound, 2 to 3 cups of properly brewed tea per day. But preparation method is not a minor detail. Water temperature is the critical factor. EGCG is thermolabile. It degrades rapidly at boiling temperature. 100 degrees Celsius destroys a substantial proportion before extraction occurs. The correct brewing temperature, 80 degrees Celsius, approximately 175 degrees Fahrenheit, below boiling. 5 minutes of steeping minimum. Bottled commercial green tea presents a separate problem. Many contain EGCG concentrations far below the biological effect threshold, some approaching 0. The tea must be brewed fresh, loose leaf or high-quality bags, correct temperature, correct duration. If the goal is compound delivery, this is non-negotiable. For those with caffeine sensitivity, green tea contains 25 to 40 milligrams per cup, substantially less than coffee. Tolerance is generally good before early afternoon. 5 compounds, 5 distinct molecular mechanisms. The question I am most consistently asked by patients who understand this research, not which foods, but when. Your body does not process these compounds in a static environment. Immune surveillance, cellular repair, gut microbiome metabolism, all follow circadian rhythms. Consuming specific compounds at times that align with biological peaks is not a minor refinement. It affects bioavailability and downstream biological effect, sometimes significantly. EGCG is most efficiently absorbed in a fasted or lightly fasted state, making morning consumption, before or alongside a light breakfast, the optimal absorption window. Sulforaphane activates NRF2 pathway gene expression, requiring several hours to fully manifest, making midday consumption well-timed relative to afternoon cellular repair activity. Beta-cryptoxanthin from papaya is fat-soluble. Fat-soluble compounds require dietary fat for optimal absorption. Even a small amount, olive oil, avocado, nuts, makes a measurable difference. Pomegranate's elagitannins are processed by the gut microbiome overnight during peak microbial metabolic activity. Evening consumption optimizes urolithin production and Akkermansia proliferation. Purple potato anthocyanins reach peak plasma concentration approximately 3 to 4 hours after consumption, making an evening meal timing well aligned with overnight repair processes. These are not rigid rules. They represent the current best understanding of aligning compound delivery with the biological rhythms that determine its effect. For anyone approaching this as a serious long-term protocol, which I would argue everyone over 60 should, the timing layer is worth incorporating from the outset. Let me give you the framework I would describe in a clinical consultation: specific, sequenced, and realistic for someone in their 7th or 8th decade. Morning, fasted or light breakfast state. 2 cups of green tea, brewed at 80 degrees Celsius, steeped 5 minutes. EGCG delivery at peak absorption. Mid-morning, 1 cup of ripe papaya alongside any meal or snack containing dietary fat. Beta-cryptoxanthin absorption maximized through the fat-soluble carotenoid pathway. Midday meal. Broccoli, stem included, lightly steamed 3 to 4 minutes, or broccoli sprouts added raw. This is when NRF2 activation begins. The sulforaphane dose delivered at midday initiates gene expression changes that continue developing across the afternoon hours. The genetic machinery responds and keeps responding. Afternoon. A 3rd cup of green tea, extending EGCG exposure into the second half of the day. Continued angiogenesis pathway inhibition throughout the afternoon. Evening meal. Purple potato as the primary starch, prepared the previous day, cooled overnight, served at room temperature. Anthocyanin dose targeting cancer stem cell pathways. This simultaneously maximizes resistant starch content for gut microbiome support. 2 mechanisms, anthocyanins and resistant starch, operating from a single food preparation choice. Before sleep. 8 ounces of pure pomegranate juice, diluted 50-50 with water if blood glucose is a consideration. This initiates overnight L-gitanin processing. Urolithin production begins while you sleep. Akkermansia population expands. The mitochondrial signaling pathway is supported throughout the night by this compound timing. Absolute daily adherence is not required. Consistent intake over weeks and months is the variable that correlates with measurable biological change, not perfect daily execution. One question I would ask anyone hearing this for the first time: how many of these 5 compounds were already present in your regular dietary pattern? In my clinical experience, the answer is rarely more than 1 or 2. That gap is precisely what this protocol is designed to close. I want to be precise about something, because precision matters in this field more than in almost any other. The compounds I have described today do not cure cancer. I want to state that without qualification. They do not replace surgery, chemotherapy, immunotherapy, or radiation. Any source that tells you otherwise is not representing the evidence honestly. I would encourage you to treat such claims with significant skepticism. What these compounds do, and what peer-reviewed literature documents with increasing clarity, is modify the biological environment in which cancer either develops or fails to develop. They shift the molecular conditions inside your cells in directions that make cancer progression measurably more difficult. That is a precise and meaningful claim. Here is what changed my thinking most significantly over 20 years. Early in my training, the dominant model of cancer was essentially deterministic. You either had the mutations or you didn't. The environment was secondary. Lifestyle was peripheral. That model has been substantially revised by epigenetic research. What we now understand, the same genetic background can produce radically different cancer outcomes depending on the regulatory environment surrounding those genes. 2 people can carry identical tumor suppressor gene sequences with completely different expression rates based on epigenetic factors that are meaningfully modifiable. Diet is one of the most significant modifiable epigenetic variables we have identified. After 60, the cumulative epigenetic drift of a lifetime begins to matter more acutely. Tumor suppressor genes fully active at 40 may be partially silenced by 60, not because the gene changed, but because the regulatory layer has shifted across decades. Sulforaphane most directly, EGCG and anthocyanins more broadly, each demonstrated the ability to interact with that regulatory layer and restore expression to genes that have gone quiet. This is the finding I consider most significant in the oncogenetic nutrition literature. Not that food contains antioxidants, not general inflammation reduction, but that specific, identifiable molecular compounds can reactivate specific, identifiable defense genes through documented epigenetic mechanisms in living human tissue with measurable outcomes. Your genome is not a fixed sentence. It is a dynamic text, and what you eat is, in a very real molecular sense, part of the editing process. Let me summarize the 5-compound framework, not as healthy eating tips, but as a molecular protocol that took decades of research to assemble. L-agitannins from pomegranate, restoring mitochondrial function through urolithin production and feeding the Akkermansia population that underlies immune competence. Anthocyanins from purple potato, selectively targeting the cancer stem cell survival pathways that conventional treatment characteristically fails to reach. Sulforaphane from broccoli sprouts and stems, activating the NRF2 transcription pathway and reawakening tumor suppressor genes that epigenetic drift has silenced. Beta-cryptoxanthin from ripe papaya, accumulating preferentially in pulmonary tissue and modifying cancer cell signaling pathways in tissue carrying decades of oxidative burden. And EGCG from properly brewed green tea, disrupting angiogenic infrastructure, boosting natural killer cell activity by a documented 47%. Together, epigenetic layer, mitochondrial layer, immune layer, cancer stem cell layer, vascular infrastructure layer. That is coverage across the full architecture of cellular cancer defense. Each of these mechanisms has been documented in peer-reviewed research at a level of specificity simply not available 10 years ago. The science has matured. In the comments, tell me which of these 5 compounds were already part of your dietary pattern, and which mechanism surprised you most. I read these responses carefully. If this analysis was useful, share it with someone over 60 paying attention to their health. The research exists. Most people have not encountered it at this level. Subscribe if you want to continue at this depth. There are mechanisms we have not yet examined: mitochondrial dynamics, telomere biology, immunosenescence. These conversations will continue. Everything discussed here represents general scientific information from published research literature. It is not a clinical recommendation or a substitute for individualized medical care. Your diagnoses, your medications, your treatment history, these determine what is appropriate for you specifically. No general protocol replaces that individual assessment. Before making any dietary changes, particularly if you are currently undergoing cancer treatment, please consult with your treating physician or a qualified specialist. That consultation is not a formality. It is medically essential. Your physician needs your complete medical history before any protocol modification. No general protocol replaces individual clinical assessment. What applies broadly may not apply to your specific biological context. Please act accordingly. Your genome is not your destiny. What you eat is part of the ongoing conversation between your environment and your DNA. After 60, that conversation deserves your full attention. The science is there. The compounds are accessible. The decision, as always, is yours.